this mini lecture, I'm going to talk about your basic setup for next week. You should have watched the video previously on a little bit about diffusion and osmosis. So this one I'm just going to concentrate on setup and basically what your hypothesis should be and what you're going to be focusing on. So basically what you guys are going to be doing, you're going to have a bunch of different solutions. So you're going to be given 1 molar of sucrose, 1 molar of glucose, 1 molar of NaCl, 2% of starch, and of course water. And with osmosis and diffusion, there are a lot of different labs that we could do. We could look at the rate of diffusion over time. We could look at how rate is correlated with the amount of the solution. What we're basically looking at for this lab is looking at a correlation between permeability and molecular weight, so molecule size. What you guys are going to do first is calculate out the molecular weight of sucrose, glucose, salt, which is your NaCl, and starch and water. Okay, all of that will be in your first data sheet. And then what you guys are going to do is you're going to think about your last lab. So the one before the cell lab on molecules. And how would you know if glucose or starch moved in and out? So what tests did we do for that lab? As far as your salt, you're going to be using a conductivity probe. Basically, a conductivity probe measures the concentration of ions in a solution. We're going to be measuring sodium chloride. And so that's going to be producing these ions when it's dissolved in water. So it should give you a reading whether or not it's diffusing through the actual membrane. We're going to be using dialysis tubing. We're going to be using 10 centimeters of dialysis tubing. This is important. That's not written down anywhere, so make sure you write that down somewhere. We're also going to be using out of all of these for each of your tubes. So you're going to be making a tube. So inside of here, you're going to have 5 milliliters. Okay, so make sure that you write that down. All right, let me erase all of that. Okay. So what you need to talk about with your group is how are you going to set up the experiment to figure out if there is a correlation between permeability and molecular weight. The larger the molecule structure, the slower it's going to diffuse through your actual membrane. Okay, you're going to be using beakers, and you're going to be setting up 5 scenarios. 4 of them, these 4, are going to be your, whatever you decide to pick, and 1 will be your control. And you guys need to talk about what you're going to be putting into your control. Okay, so you guys are going to have your tubings in each one. Remember, there are going to be 10 centimeters measured. Here we go. And they're going to have 5 milliliters inside the tubing. Don't forget to weigh before and after. And then think about why would that be important? Why would you need to know the weight before and after? You know, will your bag be heavier, lighter, or the same? Why? So these are some of the questions that you should think about. All right. And then what you're going to do once you're all done, after you weigh them, is you're going to take out your dialysis tubing, place it into another little beaker, and you're going to test the inside amount for your unknown, or you're going to test the outside depending on what was in the bag and what was on the outside. So again, this is all up to you guys. Alright, if you have any questions, let me know. Again, make sure that you watch that first video on osmosis and diffusion, and I'm also going to put up a little video on the conductivity probe. One thing I did want to mention for the conductivity probe, in case I do forget to mention this in class, so for the probe, you are going to set your probe to 0 to 2,000 microseconds per centimeter. Okay, so on your actual probe, so let me erase this real quick. Oh, come on. Okay, so on your actual probe, so you have your LabQuest. I'm going to give you guys a quick little demo on how to use the LabQuest. Okay, and then attached to your LabQuest is going to be your conductivity probe, and it's going to have a sensor right here, and that's what's going to detect those ions. Okay, and there's going to be a box right here. On this box is going to be a switch, and this is the switch that's going to say 0 to 2,000, and that's going to be your range. Okay, you're going to not keep, now the video I'm going to show you about the conductivity probe has you keep the probe in there for the entire time. We're not going to do that. You guys are going to do a test before and a test after. Okay. Also, for your testing for your glucose, we will not be using the Benedict's test. We're actually going to be using test strips, which run on the same principle, and I may have talked about these in your class. So the test strips will turn a certain color depending on whether or not it's positive. Alright, that is it. I will see you guys next week.